So we have here a cell that is ready to undergo meiosis. We have our chromosomes paired with their sister chromatids. So what's going to happen is they're going to line up along the center line as the homologous chromosomes will line up in pairs just like this here. Okay, so they'll line up in the center just like this. Then once it's made sure that each of the cells is going to end up getting one of the homologous pairs, we will undergo our first cell division in meiosis 1, and they will move to opposite sides of the cell during anaphase 1, and then of course we will have telophase 1, and we will separate the two. So now we have one of the daughter cells from meiosis 1. So again, we're going to line our chromosomes up along the center just like we do in mitosis. However, now we essentially only have about half of the unique genetic material. So now what we'll do is, just like in mitosis, separate the sister chromatids from each other and pull them to opposite sides of the cell during anaphase 2. Then, once they're sufficiently pulled to opposite sides, we'll undergo telophase 2 and cytokinesis and separate our two daughter cells, giving us our first type of gamete. So now we have the other daughter cell from our first meiosis 1. So again, just like we just did with the red ones, we're going to line them up here along the metaphyseal plate in metaphase 2. Again, separating our sister chromatids just like we do in meiosis or mitosis. In anaphase 2, we'll pull them to opposite sides of the cell. And then in telophase 2, again, separate the two, giving us our other type of gamete. So now, of course, as we know, how the chromosomes line up initially is based on random chance. So there we organized and kept our chromosomes of the two colors together. But of course, the assortment of the different chromosomes is going to be completely random. So, again, making sure to line up our homologous pairs on either side. We now have a different initial setup for meiosis 1 that's going to give us different gamete cells than we just got from our last meiosis example. So again, we're going to pull our chromosomes and separate our homologous pairs from one another in meiosis 1 and anaphase 1, go through telophase and cytokinesis 1, and separate these two into two different daughter cells. So now we have our first daughter cell of our second look at meiosis. So again, notice the difference here, how we have the yellow big chromosome and the red small chromosome. That's different than the setup that we had last time. So now as we go through meiosis 2, we're going to line up our chromosomes all along the center here. We'll separate the two sister chromatids just like we do in mitosis. Pull them to each to different poles of the cell. Then in cytokinesis and telophase 2, we'll separate them, giving us these two gametes. So now we have the second daughter cell of our second look at meiosis. So again, we'll line up our chromosomes along the center line here, separate the sister chromatids and pull them off to one side. Then we'll undergo our telophase 2, cytokinesis, and separate these into our other type of gamete possibility. So sometimes we can have errors or mistakes that can happen during meiosis. Depending on which stage of meiosis they happen, meiosis 1 or 2, they can have different effects on the gametes that are produced from that meiotic division. We're going to be taking a look at non-disjunction, looking at what happens to our gamete cells at the end if we have a non-disjunction event, that is a failure to properly separate chromosomes, in meiosis 1, also called non-disjunction 1. So normally in meiosis, right, we line up our chromosomes along the center, And normally we separate our homologous pairs from each other so that they move to opposite sides of the cell. However, with non-disjunction, this would fail to happen. So for the majority of our chromosomes, they'll properly separate the two different sides. With non-disjunction 1, we will end up taking both of these homologous pairs off to one side. Then, when we undergo cytokinesis, what we end up with is one cell here that is completely missing a specific chromosome, and one here that has double that number of chromosomes. So now we have our daughter cell of that non-disjunction 1 event. So again, we still have these two copies of the homologous chromosomes here. But in meiosis 1, all of the chromosomes line up in the center line just like they do with mitosis. So we now no longer have an opportunity to separate those two pairs of homologous chromosomes. And as a result, assuming meiosis 2 goes normally, we'll separate each of these chromosomes into their sister chromatids and pull those to the opposite portion of the cell. So now when we undergo meiosis 2 and undergo telophase 2 and cytokinesis, what we end up with is these two gametes having an n+1 number of chromosomes, because instead of each of these cells having 2 chromosomes, each of these cells have 3. So here we have the other daughter cell from our non-disjunction 1 event. This one here is missing an entire chromosome, right? It's missing that whole set of sister chromatids. So when it undergoes meiosis 2, it lines up, in this case, the only chromosome that it has, splits them into the sister chromatids, and then undergoes cytokinesis and divides the two. As a result, we now end up with an n-1 gamete, because instead of having the 2 chromosomes that should be present, it only has 1. So now we're going to look at what happens if there's an error in meiosis 2. But first we're going to start off with showing what would happen in the regular meiosis. So our meiosis 1 is just going to proceed normally. We'll line up our homologous pairs along the center line, and then we'll separate them each to a different half of the cell, and then divide those two cells, giving us the two daughter cells of that first meiosis. Everything here is functioning normally. So in the next step, where things are going to go wrong. So in this daughter cell, we're going to replicate what happens in a non-disjunction 2 event. So normally what happens is we separate our sister chromatids, but with non-disjunction, we fail to have that proper separation. So for our big chromosome here, it's going to take both of the sister chromatids into this other cell here, and our small chromosome will be able to split as per normal. Then when we undergo cytokinesis and separate these into 2 different gametes, what we can see now then is that we have an n+1 cell here and an n-1 cell here. But what's different about this n+1 cell from a non-disjunction 2 event versus a non-disjunction 1 event is notice that both of these chromosomes here are the same color. These are genetically identical. A non-disjunction 2 event will give you 2 genetically identical copies. Non-disjunction 1 will give you 2 different copies. So now we're going to look at what happens in the other daughter cell for a non-disjunction 2 event. Now, non-disjunction events are very rare. So if you do have a non-disjunction 2 event, it's very unlikely that it's going to happen in both daughter cells of meiosis 1. So when we undergo meiosis 2, for this daughter cell, it's going to end up looking exactly like we would expect for meiosis 2 with this given chromosomal composition. So, whereas non-disjunction 1 will give you 2 n+1 cells and 2 n-1 cells, non-disjunction 2 gives you an n+1, an n-1, and then 2 normal n cells.